Hi, everyone. So my name is Daniel Mobilio, and I'm going to be talking about my final research presentation that I've completed through the Brain Tumor Foundation of Canada summer studentship. And I completed my research under the supervision of Dr. Sheila Singh. She's a pediatric neurosurgeon and a scientist who studies new therapies for brain cancer. And specifically, I study in her lab brain metastases, which is the spread of cancer from somewhere else in the body to the brain. So I'll just quickly first delve into that. So brain metastases are the most common form of CNS malignancies. So what does that mean? It's the most common form of brain cancer worldwide. And so frequently brain metastases arise from the lung, breast, and melanoma cancers. So the lung, breast, and melanoma, if someone unfortunately, if cancer arises within that organ, they have a high propensity of going to the brain. And so brain metastases are 10 times more prevalent than primary brain tumors. So that's when a primary brain tumor is when a tumor arises sporadically within the brain. And as I said, lung, breast, and melanomas have the most frequent spread to the brain. And of course, brain metastasis is associated with poor prognosis and survival rates, as the median time of survival for patients is 4 to 6 months. And so this really inspires our lab to create new therapies to provide hope for the future of these patients. And so that's what we work on in this lab. So to make new therapies, we first have to understand how does brain metastasis work. So this diagram that I have here on the left side of the screen, it's called the brain metastatic cascade. So this is the process of cancer cells that are in somewhere else, is in an organ somewhere else in the body, and they're able to invade out of their location and get into the bloodstream, where they're able to then migrate through the blood system and find themselves in the brain, where they create this secondary tumor. And so, as studies have shown, only 0.01% of the cells that are in the primary tumor, let's say it's the lung or melanoma or skin or breast tumor, only a very small percentage of these cells actually make it to the brain and create a full brain metastatic lesion. And so when you look at the primary tumor, we could kind of characterize the cells within that tumor in two different ways. So there are these small populations of cells called cancer stem cells that we study in our lab. And then there's these non-tumor initiating cells that represent most of the tumor. And so the most of the tumor is able to be eradicated by chemotherapy or radiotherapy, the usual cancer treatments that a patient would go through. However, these cancer stem cells that are a small population within the primary tumor are able to self-renew, proliferate. They create tumors through tumor regenesis. They're resistant to therapies, and that leads to recurrent tumors, or in our case, it leads to brain metastases. And so this capability of self-renewal is the ability of cancer stem cells to able to repopulate a tumor, and the proliferation is the capability of these cells to be able to proliferate and create new tumors and whatnot. And so our lab is focused on actually targeting the cancer stem cells, as we believe that these are the cells that we need to eradicate in order to try to find better treatments or hopefully cures for cancer. And so in our lab, we've discovered these brain metastatic initiating cells, or these cancer stem cells, and we call these BMICs for short. And so to actually study these BMICs, what our lab does is we are fortunate enough to receive patient donations from right after their surgery, and we bring those samples to our lab and we culture them in the correct conditions to enrich for these cancer stem cells. And so now we're studying specifically the brain metastatic initiating cells in our lab. And so just to reiterate what I presented last year, was the results from my first summer internship at the BTFC. And so the goal of the first summer was to develop a migration assay to quantitatively and quantitatively identify the brain metastasis migration capacity. And so basically the simple question is, are we able to model this part of the brain metastatic cascade in a dish? And so after the summer and through the summer of a lot of work, we were able to develop a migration assay. And I talked about this last year, how you have to optimize for cell numbers and you have to do specific imaging techniques to understand the migration capacity of these cells. And you're able to generate some videos and photos such as this one. And then you're able to have data such as this. And so the second goal of the BTFC studentship that I was pursuing was aimed at determining new drugs that can impact brain metastases in vivo and in vitro. So that means, can we find new drugs that are able to target brain metastasis in a dish, and then if that works, move to animal models. And so I won't get into the nitty-gritty detail of how we discovered dihydroortic dehydrogenase as a hit or a target in brain metastases, but I'll just quickly talk about DHODH and its role as an enzyme. And so DHODH is found in the inner membrane of the mitochondria, and it helps in the production of uridine. It's one of the major steps in the production of uridine, which is very essential for all cells. And also its secondary function that doesn't have a lot of research that goes into it is that in its reaction of making uridine, it shuttles the electrons from complex 2 to 3. And so in this project, we were interested in understanding how DHODH is important in producing this uridine for the cancer cells, but also in its secondary function of helping the mitochondria function correctly. And so our simple question, I guess, to show that this works is, can we use this drug in an in vitro system and see that the cells die? And so through these three graphs here, the top two and the bottom left, the lower that the slope goes in the graph is the more cell death that we have after we use the DHODH drug. And so with these patient brain metastasis lines that we have in our lab, we tested them and we saw that this drug works very, very well at targeting brain metastases. And so secondly, after we start to discover that this is a hit or a target in brain metastases, we want to understand the expression of this enzyme across different sensitive cells in vitro. And so this is just the data here. And the key part of this experiment is to show that the cancer cells are actually expressing what we're trying to target. And it starts to show that we are targeting something that's actually there and that the drug is working as it should. And so this is just a Western blot where you're able to quantify a certain protein of your interest. And so in the top panel of the Western blot on the left-hand side is the amount of DHODH. So the larger the fluorescence or the more dark the band is the more you have of the enzyme. And then vinculin is just a control protein that we use to gauge if we messed up the experiment or whatnot. And so when you compare the expression level of DHODH to how sensitive the brain metastasis cells are, there doesn't seem to be kind of a correlation as something to highlight, say the second cell line BT673 which is a melanoma to brain metastasis cell line, is very sensitive to our drug in the nanomolar range. However, it doesn't really have a very high expression of DHODH. So these are one of the questions that we want to try to answer in the future that we haven't got to in the summer. However, it is very interesting that a known drug that inhibits DHODH and it really impacts these cells, they're doing so on a cell line that doesn't express DHODH so that like very much. And so could be some very interesting very interesting reasons as to why this inhibitor is so potent. And so one of the main objectives of our lab is to find therapies that not only kill cancer cells, but are safe and effective. And so in our lab, we grow healthy tissue. Usually it's neural stem cells, which everyone has within their brain, even when they're older, and normal human astrocytes, which are found all over the brain. And so we culture these in vitro or in the lab, and we use our new therapies on them to see if they are affecting the normal tissue. And it kind of gives us a guideline into understanding if the drugs that we're finding or new therapies that we're developing is specific against the cancer. And so that's what we saw through this data, where the neural stem cells and normal human astrocytes were not affected. And something really interesting to note is that these graphs here, or these dose response graphs seen on this slide, these are in the micromolar range, as if we quickly go back to the dose responses here, which they're in the nanomolar range. So this drug was working very well against the cancer in a very low concentration, whereas even at a high concentration, it doesn't affect normal tissue or normal cells. And so this data really excited us to keep going further into developing potentially a new drug or potential treatment for brain metastases, as it seems to be very potent and safe. And so to understand how well your target actually is, a lot of researchers, they do genetic studies. So all the studies that I was just talking about before, that was a pharmacological type of study where you're using a drug to pharmacologically inhibit an enzyme. But to further prove that the gene or enzyme that you're looking at is important in cancer, is that you genetically get rid of the gene and see what happens to the cancer cells. And so that's what we did next, was to genetically understand if we get rid of this enzyme or this gene in cancer cells, what happens. And so this data just shows quickly that with different types of CRISPR, we're able to successfully get rid of the enzyme in the cancer cells. And so with these cancer cells that have this gene knocked out, we do a bunch of functional assays. And so what that means is we test different types of capabilities of the tumor cells or cancer cells to understand how the genetic deletion of an enzyme impairs the functionality of the cancer cell. And so this experiment is a sphere formation assay, and this is a surrogate of self-renewal. So it's able to test the self-renewal of the cancer stem cell or the BMIC. And so, as I said earlier in my presentation, self-renewal is very important when we look at new treatments of cancer, because self-renewal is a driver of relapse and it's a driver of brain metastases. And so through this experiment, we understood that if you genetically get rid of this gene in the cancer cells, they lose their ability, or their ability to self-renew is very hindered. And so this was a nice functional assay showing that this gene is very important for these cancer cells. And so next we did these proliferation assays. Proliferation assays just understanding how fast that these cancer cells are replicating. And we saw with these functional assays that when you get rid of the gene from the cancer cells, the ability of these cells to proliferate was significantly attenuated. And so now we found this evidence of showing that important processes in brain metastases, such as self-renewal and proliferation, when you get rid of this gene, is significantly altered. And so with this data, we wanted to do an in vivo knockout trial. So what that means is that you knock out the gene and then you insert those cancer cells into the brain of a mouse, and we then model this disease in an animal, and we try to understand how a single genetic knockout or a genetic deletion of a single gene impacts the cancer in an animal model. And so through this data, we saw that mice with one of the CRISPR knockouts significantly lived longer than the controls. And so this was the first data to show, at least in vivo, that's very important. This gene is very important to the progression of brain metastases. And so one thing I would like to highlight here is it may not seem like a big difference between the red and black lines. So the black line, AVS1, is the control, and then the red line is the knocked out cancer cell, and the yellow is a different type of knockout. It's the same gene, but just was done differently. And so this doesn't seem like the biggest difference. However, since our model is only 30 days, an extension of like a week or 2 weeks that we see here is actually quite significant. And so we understood genetically in vivo and in vitro what's happening with the mice. And so the last experiment that we conducted in the summer was to understand how does pharmacological inhibition affect the cancer cells in vivo. So can we use this? So basically what that means is, can we use this drug, give it to the mice, and can they live longer? And so to do this type of experiment, you have to intracranially inject the tumor cells into the mouse's brain, and then daily we orally gavage them, or we administer the drug orally we just deliver the drug orally, and then we would understand how the tumors progress through IVIS, which is like an in vivo imaging system. So you're able to understand how the tumor grows and changes as the mice is alive. And so through all these two different drug trials with two different DHODH inhibitors. So on the left is BA240-2234, which is a DHODH inhibitor, and a different DHODH inhibitor, BTC868. When we orally gavage the mice or administer the drug every day to the mice that has the brain metastases in them, they lived longer. And so this was maybe one of the most key findings, as it shows that a mouse with a brain tumor can live longer if you give them this drug. And so for future plans of the project, we want to understand how the mechanism of DHODH inhibition in brain metastases. So what does that mean? We want to understand why inhibiting DHODH is cancer selective and it's very potent, and why does it work so well in brain metastases. And so DHODH, as I mentioned, is a part of metabolism, as it creates uridine, and it also plays a role in the function of the mitochondria. And so we also want to identify, why do the cancer cells rely on DHODH, and things, and answer questions such as those. And so lastly, through this experience, I gained such an amazing love for science and desire to keep going forward. And now I'm pursuing my PhD in the Sheila Singh lab, and our future plans for this PhD is to explore new possible treatments for brain metastases, not only DHOH, but other things. And for that, I would like to thank the PTFC and the Tate Boomer Foundation for supporting me through this studentship, as it inspired me in the career that I've chosen. And so, yeah. Thank you very much for listening. And those were my two, that was my research for the two research summers under the BTFC scholarship.